Thank you everyone for coming. It is my great pleasure to introduce our today's speaker, who is our colleague, collaborator, and friend from here at NYU from Quran. It's Professor Mike Shelley. He is a Lillian and George Little Professor of Applied Mathematics here at NYU, as well as Professor of Mathematics, Neuroscience, and Mechanical Engineering. Mike is one of the founding fathers of the Applied Mathematics Lab in Quran that many of you are familiar with. He is currently one of the serving co-directors also. He is director of the Center for Computational Biology in the Flatiron Institute at the Simons Foundation. And to give you a little picture about his trajectory, about his path, so as you can imagine, Mike is a mathematician by training and by the trade. He got his BA in math at University of Colorado, PhD in applied math at University of Arizona. He completed his postdoc at Princeton before he joined briefly math faculty at U Chicago before coming to NYU, right? So he has received for his work a lot of highly prestigious prizes and awards. Examples of those, for example, Francois Frankel Award from APS, Julian Cole Lectureship from the Society for Industrial and Applied Mathematics. He is fellow in both of those societies, so APS and CM. He is also fellow of AAS and member of National Academy of Sciences. His research focuses on modeling and simulations of complex systems in both physics and biology. And as you will see in his talk, he particularly enjoys self-organizing processes in biological systems, which he addresses using tools for mathematical modeling. He develops numerical tools, he uses analysis and large-scale models. So without further ado, Mike.
Introduction, maybe. Thank you. So I haven't. I think the last time I gave a talk here was maybe 10 years ago, and I was talking about active suspensions, bacterial suspensions, like one of the prototypes of active matter. And I've gone on from there to other things that were strongly influenced by that work. And today, what I'm going to tell you about is I'm not going to be talking about nuclear today. My joint work with Alexandra. And but I'll be telling you about dynamics of development inside of large cells. And the large cells I'll be talking about are embryos and egg cells, or oocytes. And the fact that they're large, that stems from the fact that they have to develop a whole organism, sometimes multicellular, from all the components that they have to be containing within them. And because they're large, they can have rather special transport mechanisms for getting things around in the face of the inefficiency, maybe of diffusion, that kind of thing. Okay, so here's my list of collaborators. I want to point out my longtime collaborators at Flatiron, Reza Faharapar and David Stein, who are in the biophysical modeling group. Dan Needleman that we share with Harvard and CCB, Center for Computational Biology, and Stosh Schwartzman, who we also share. A lot of the people from Princeton, a lot of the people that we work with are experimentalists, including Dan, and Stosh has a hand in that as well. And there's my blur. Okay, so let's talk. I'm just going to start with the very first cell division in a very highly studied model organism, which is C. elegans. It's this small 1 millimeter worm. Has 957 cells or something like that. It's a very prototyped organism. And what you're seeing here, you can see the eggs inside of there actually, but what you're seeing here is a egg that has just been fertilized, and those big blue blobs are genetic material. They're called pronuclei from the male and the female. The male has come in at the right, and it has arrived, and a lot of this dynamics is kind of stereotypical of many other organisms. But the male pronuclei has arrived with these two bright spots, which are called centrosomes. And these are sites, small organelles, that are nucleating microtubules, so rather stiff, relative to thermal fluctuations, rather stiff polymers that grow just through end polymerization, and they can support loads and exert loads, and they do a lot of the force transduction that you need to do in order for the kind of dynamics that you see here to happen. And so what's the dynamics? The male and female pronuclei have come together. This complex rotates to the long axis of the cell. Chromosomes have divided. And then at the end of that movie, you will see the cell membrane start to come down. We stop short of actually showing the cleavage and the formation of two new cells. And you can see how structured, this is just light microscopy, so a lot is washed out, but you can see these kind of spots of like a aster, they're called asters, these kind of star-like arrangements of microtubules shooting out. So this is, as I said, very much studied. And I'll talk a bit more about this later, but except to say that what is driving all of this is another workhorse of the cell, aside from microtubules and centrosomes, and those are motor proteins. And so it turns out that the boundary of this cell is coated with several hundred, at least, what are called dynein motors. And dynein motors are little molecular machines that have evolved to bind onto a microtubule and walk towards the centrosome. So microtubules are polar, they have a direction. Their plus ends come out and polymerize out into the cell from the centrosome. And dynein motors like to attach and then walk towards the minus end, which is embedded inside of the centrosome. So while all this is going on, this is all taking place inside of a cell. It's packed with an aqueous slurry called the cytoplasm. It's very complicated, but it flows. How was your talk? How was your talk at Garage? I didn't get to go.
It was lovely.
Okay,
I had a great
great.
time. They told me I was full of shit.
Who told you?
As mathematicians do.
Okay.
Payback time.
Here I am. Okay. And so there's flows inside of this as all this is going on. And what you're seeing here is actually a particle imaging velocimetry done on endogenous, actually no, they're not endogenous, on little fluorescing nanodiamond particles that have been injected into the parent worm. They get taken up by and this is done in Dan Needleman's lab. They get taken up into the progeny, into the eggs, and you can use them as fluid markers. And so while all this is going on, there are flows going around inside. And it turns out you can use those flows to try to figure out how forces are actually being transduced to have all this kind of dynamics going along. So that's an example. Fluid mechanics inside the cell, and here maybe used as a signature of something. Now, when I talked about centrosomes and mitotic spindles and asters and all this kind of stuff, as a modeler, so my background is in computational fluid dynamics. I started off in high Reynolds number flow, and then slowly degraded myself over time until I'm down at Reynolds number 0. But so what you're seeing here is actually from electron tomography. So this is a flash frozen cell that is then put into an electron microscope and reconstructing all the microtubules within that cell and other things you may want to see. And in C. elegans again, so this is actually showing you that object that you saw kind of moving around at some stage. This is the mitotic spindle here in the center. Those are the chromosomes, and these are microtubules that are going out and touching or binding onto these motors on the boundary. So it's geometrically very, very complicated. After that, the cell divides. And it just keeps doing the same thing. What's interesting about something like a nematode is that you have this egg, and it divides into 2 smaller cells. There's another division, 4 smaller cells. You keep having the volume more or less, but the same thing just keeps happening over and over and over again. And so you need to have a robust mechanism for making sure that you can divide the chromosomes at these stages where volumes and sizes and all these kinds of things are constantly decreasing. So that's another interesting thing to think about.
Mike?
Yeah.
Can you orient us a little bit on any scale here? Like what What is
sure,
the
I'm
size
sorry.
of
Yeah.
one of these?
So this is about 50 microns.
Okay.
Yeah, thanks. About 35 in this direction, something like that.
What's the time step for the video? Like how?
I mean, what's the time scale?
Yeah.
So for this first video, the time scale was for about 15 minutes, something like that. I'm unsure about this, actually. That's not mine. So I don't have to, I just get to use the advantages and not be responsible. Okay. But that's what happens. Okay, so then this is the the kind of obligatory what are all these kind of functional components that are inside of cells that are doing work. And so that's what's called the cytoskeleton. So it's a set of cross-linkers and polymers. Some of the cross-linkers are active, some are passive. The active ones move the polymers around or attach onto other things and use the polymers to move things around. And so this is actually a fluorescence labeling of 3 different important polymers inside the cell. The green ones are those microtubules I was talking about. They tend to be rather stiff. They have persistence lengths that are well beyond the size of the cell. Then there is actin, which is the red stuff. It's a shorter, more flexible polymer that's used as well to kind of transduce forces or to give rigidity to objects within the cell itself perhaps. And then that's DNA, which we don't tend to think about mechanically, but more in an information theoretic sense. Okay, for what I just showed you, just to set some physical constraints, inertial forces are completely irrelevant in this case. If you form a Reynolds number, people talk about the Reynolds number, oh, bacteria 10 to the minus 5. They have nothing on the cell. So Reynolds number here is down around 10 to the minus 8. You're confined, which means you go slower because you have the slow slip condition on the boundary at the least, if you think of it as a fluid. For what I just showed you, you take the scales, 10 to the minus 8. This is what I was saying, you can be very sensitive to confinement. Forces scale badly with having to do things in smaller and smaller volumes. And if you look at the elasticity of this kind of aqueous slurry, which you can see, it's just packed with stuff. But another interesting question, which I have no answer for, but someone brought this up, so it's not my idea, is that the cytoplasm is very complicated, it's full of stuff, but it has to be kind of tuned because certain processes have to take place, so it's got to give way. And so if you look at what's called a Weisenberg number, so a relaxation time times the strain rate, that tends to be small. So you could think of it as being some approximation of viscous response. And I'm not going to talk about force dipoles today, I think. Okay. I just want to show you another example, just because it's fun. So that was light microscopy. Well, it's all light microscopy, but it was a living cell. Maybe it was excised, but it's kept in some medium for a while you watch all this stuff go along. And so people play games in synthetic environments where they use the fluidic kind of purified's the wrong word, the extracted cytoplasm that contains all the ingredients you need to do things like grow microtubules and all that. And they will put in a synthetic centrosome. It's coated with a protein that causes microtubules to nucleate. And then they look at the dynamics of things in kind of these more controlled geometries. You could knock down things more easily. You could measure things more easily. And I just like this one. This is a set of experiments from a lab that we collaborate with, a guy named Jesse Gatlin, University of Wyoming. And what they do is they take what's called the extract of the cell, they put an artificial centrosome into it, and they just see what it does. And here there's no motors. They've been knocked down. Okay, so there's no motors. And so what you see here is that the microtubules grow out from this place, and they just push on the boundary. Maybe they buckle. You can see kind of buckling inside of here. The green is EB1, which is a tag for the growing plus end of a microtubule. And what it nicely does is it just centers itself. Centering of something is very important. Finding a position is very important. It's kind of a general principle in cell biology. But it does other things that you don't see in cells typically. Once it's centered, it just starts to rotate around. It's a symmetry breaking instability. It will go both ways. And if you make the cell a little bit bigger, it just goes back and forth like a washing machine, actually. So in these kind of synthetic environments, you can learn things about how these objects would behave if you pull them out of a highly regulated environment of the cell. So I think that's interesting. Yeah.
Does it go back and forth stochastically or deterministically? And the latter, why?
Well, in the experiments, you're right, I've looped it. So I'm being slightly dishonest. Slightly. You can get 2 or 3 of what look like things, oscillations in the experiments that are very slow and you kind of run out of juice after a while. You'll get 2 or 3 oscillations that look like they have a nice period. On the other hand, we have a model that gives you a nice oscillation directly out of it. So we think it's deterministic.
And how much bigger is this than a cell? How much bigger is this
Oh,
apparatus
good question.
than a cell?
So this is a very large cell. This one I think is around 50 microns across, and this one is 80. So this would be on the size of a oocyte, or like an egg cell, really. But in our world, it's still pretty small. It's not a cell.
It's really not a sailboat. But the shape of the container, like it's sad that it's a cylinder. Shouldn't you do a more interesting shape?
So the experimentalists wanted to make sure that it was a symmetry breaking instability.
I see.
So what they did is they wanted to choose a direction.
I see.
And so what they did is they put this kind of scalp on the side, and then they showed that it would only go in one direction.
Yeah.
These kinds of things. What also matters is the boundary conditions. If you do this in a droplet rather than in something with solid walls, it behaves differently. So in a droplet, all of this dynamics is really driven by the fact that if you watch enough of these movies, you realize that when a microtubule hits the boundary, it gets pinned. That doesn't happen in a droplet.
Mm-hmm.
And it's that pinning of a growing thing, which has been growing microtubule into the bulk, that drives it.
Yeah.
It's like you're hitting a wheel with a fire hose on the rim. So. Okay, this is where I'm going to spend most of my time. This is my third example. And so this is another model organism. We actually have a small lab now at Flatiron where we have incubator for flies and that kind of stuff. We had to have a couple of outbreaks, a couple of jailbreaks, I should call them. So that's the common fruit fly, Drosophila. And what you're seeing here is its machine for making eggs. Drosophila are very good at making eggs, as you probably know. And it's called an egg chamber. An egg chamber starts off as a single cell. That single cell divides 4 times to give you 16 cells. It fills this little volume here. So there's, at some point, I think this is not understood what does this, but there are 1 or 2 cells that gets chosen in this kind of lineage to be the egg. And the dark void that you're seeing here is the egg. And what you're seeing here is the nuclei and the membranes of everything else. There's a nucleus in there, but it's kind of on the back side, probably, where you can't see it in this sheet. So here it is growing, and at some point at this stage, this guy is 150 to 200 microns in length. The viscosity of cytoplasm is about 1,000 times that of water. For something on that scale, if just on pure diffusion, something the size of a protein takes about a day to diffuse across that distance. So maybe at some point in the past, the fly decided it didn't like that. And what it does is you start to see the emergence of small-scale kind of disordered flows at stage 9, and then very commonly you see large-scale kind of cell-spanning flows of the cytoplasm. That if they're there for transport reasons, then it brings your transport time down to about 25 or 30 minutes. So, and if you knock down the ingredients, they're driven by motors again. I'll be talking about a model for that. If you knock down the motors that are involved here, they're called kinesins instead of dyneins. And you can stop those flows, and then the fly will not develop properly. In particular, things need to get from these nurse cells, they're called nurse cells, which are producing things like mRNAs and things that you need for development of the organism. And for example, it needs to move them to this polar end, so-called polar end of this oocyte, of this egg. And that sets the axis of the body, and the body plan starts to get arranged.
Like in a kind of general case, does diffusion plus a circular flow like this lead to a homogeneous distribution of material?
No. Yeah, so I think this is this is why I'm kind of couching. I think this is kind of a mystery here. That if you, I'll wait till you see, and then you'll say, well, that shouldn't work very well. And then I think maybe it's not there just for transport, but for other things. Okay, here's a movie. So then this is bright field, and you're just using endogenous particles in there. And that's what one of those flows look like at stage 10. Okay, so there they are. And what is interesting about that? Okay, so you can see there's a big vortex here. There's some sort of, rising flow here. There's a stagnation point or something, but, it's pretty structured. And it varies. You take a different egg cell and it will have a different structure, but it will be on kind of the scale of the cell. The reason why it might look different is that if you have something that's very prototyped, but you have a random orientation at which you're looking at it, it's like somebody with a blindfold and the elephant, right? You're not quite sure what you're touching, what you're seeing. So so what is strange about that flow? Yes?
It looks to me like it's a wheel rather than a vortex.
Well, it looks like a solid body rotation.
That's what I mean.
Yeah. So that would be a wheel. That would be a vortex with constant vorticity inside.
Which is weird.
That's right. And why is it weird?
Well, fluids get sheared.
Well, fluids get sheared, and they get sheared because, for one thing, they satisfy typically a no-slip condition at the boundary. Right? That produces shear.
But there's no shear inside the wheel, or there's little shear. There's too little shear inside, so it's like some solid body.
That's right. But what I'm trying to lead you to my answer, which is it looks like there's a slip at the boundary. Okay, so what this is telling you is that it's got whatever is driving this. It's a surface effect. There's some sort of surface stress which is driving this thing around that allows it, on some scale, to evade the no-slip condition. It allows it to look like a solid body rotation. So we weren't the first to study this, and there was a conceptual model put forward 10 years ago or so now by the Saxton lab, and we're just gonna run with this, kind of put it on steroids. And so the picture is this. I call it conceptual because I think a lot of the microscopic underpinnings is not clear that it's got all the right stuff. It reproduces a lot of the experiments. But here's the picture. There are microtubules that, now these are known facts, there are microtubules that are embedded, whose minus end is embedded into the side of the cell. And they have grown into the interior. So the plus end is out there in the void, so to speak. So here's the schematic picture. There are motors, and they're called kinesins. And kinesin motors carry payloads. Here they're represented as little purple spheres. It could be other microtubules, it could be other things, but they carry payloads towards the plus end of a microtubule. There are kinesins that do the opposite, but these are not those. So what might happen to this? So imagine there's a little force diagram right there. I'm carrying this payload up and I'm dragging it through this fluid. So I have to exert a force on the fluid to drag this guy through it, say at a certain speed. But I'm walking along a substrate, that's my sidewalk. I have to be exerting an equal and opposite force on the thing that I'm walking up. So if I have motors carrying things up these microtubules, I am exerting a compressive load towards the base of this microtubule. What does that want to do? This is a follower force problem for buckling. It's like a fire hose. I keep saying fire hose today. I don't know why. But it's like a hose, your lawn hose. You're shooting. You can shoot water out the end of it, and it can start oscillating around because you're exerting this force on this elastic object, and the direction of the force is following the deformation of that object, and that can give you oscillations. Now, here I haven't said anything about oscillations, but there is an instability you would expect in this type of system. But the thing about it is that these are not isolated. These are immersed in a fluid, and if something moves, it moves the fluid, and that will cause other things to move. And so what I'll show you is that within this model, there is a self-organizing instability that will yield flows that look like that. Okay, I'm not going to say much about that. What do I want to say? Okay, this is just, there are many ways to model. But there are many ways to model things like that. So, as a numerical analyst or as a computational mathematician, you would say, oh, how am I going to model this system? It's like a very complicated fluid structure problem. I have all these long, skinny things that are sitting in a fluid. They're sitting inside of this volume, attached to the wall, all these kinds of things. So the first thing we're going to do is we're going to make our life really simple because we really don't know what else to do. We're just going to assume that the cytoplasm obeys a linear Stokes equation. We're going to ignore anything, any sort of geometric anisotropy from the fact you have stuff in there, and we're going to ignore elastic responses. Just say that straight out. And then you start to have a choice of methods. There's the immersed boundary method, originated by Charlie Peskin at Courant. Then Rosanna Zia, who was a student of John Brady's at Caltech. There's a whole other plethora of methods that are built around what are called Stokesian dynamics, another approximation of the Stokes equations. And then there is my favorite, which is boundary integral methods. Boundary integral methods change solving the Stokes equations in a volume. Because they're constant coefficient homogeneous equations, you can generically represent their solutions as distributions of sources and dipoles on all the boundaries, just like a Laplace equation. And so you just move to representing flows in terms of integral equations that sit on the confining geometry or on the movable objects within the fluid. They talk to each other through Green's functions. And there's a lot of people that have worked on this over the years. My work goes back to 2004 when I was working with a postdoc at Courant named Anna Karen Tornberg. And other people more lately, people that we knew or know, Alexander Donev, Andrei Maxim, and so forth, Charlie Peskin, many people. It's become a kind of an industry, a small industry, an industry nonetheless. Okay, so let me just build a model, and it's based on boundary integral methods and what's called slender body theory for the Stokes equations. How do you represent the dynamics of a long, thin object moving through a Stokes fluid? And so it basically looks like this, is that for each of the microtubules, so here's my microtubules, they have motors on them, they're exerting loads. Then this is the drag force on some little section of the microtubule, so it's written as its position, the ith microtubule x of s and t. S is the arc length measured from the base. So now S, these are inextensible objects. So S is also a Lagrangian label for a microtubule. So the motion of each microtubule relative to the flow multiplied by a viscosity multiplied by an anisotropic drag matrix is the drag force. And that's balanced by the forces that are acting upon the microtubule, which here we model as an Euler beam. And then this is our sum total model for the action of motors. And it simply says a motor exerts a force along, I keep needing to change that to an x. That's supposed to be a tangent vector, dx ds. Forces act along the tangent vector towards the base of the microtubule. We just assume it will continue. So it's not a discrete model in that sense. And then if you retain what are called monopolar forces, which are the leading order forces in the way things will communicate to each other through the fluid. It says that the flows, so that's the micro problem, the flows macroscopically are produced by summing up all the internal mechanical forces and using them to force the Stokes equation, plus any boundary conditions, like no slip or if they're attached to something else that you need to do. What are you doing on time? Good. Okay. This model has a number of dimensionless parameters. I'll mention two. So, there's like a viscosity, there's a rigidity, a length, a scale, that kind of thing. And from the parameters in that model, you can construct 3 time scales. One is the time scale it takes for an elastic object to relax from deformation, which depends on its rigidity and its length and the viscosity. A collective relaxation time. And this is the interesting one. If I go back to this macro part, all these elastic objects are talking to each other through the fluid. This sums up all those forces and gives you another relaxation timescale. And that's the one that says we move together, we relax together. And that relaxation timescale turns out to be divided by, I've written things in kind of a dimensionless way here already, but this is the areal density, the number of microtubules per unit area that are impinging into the fluid. So it's a density. The more microtubules you have, the faster that collective relaxation timescale is. So it's directly talking about the contribution of summing the forces through the Stokes equation. Then there's a time it takes a motor to carry a payload the length of a microtubule. That's another. And from these 3 timescales, we have 2 important parameters. One is a dimensionless motor strength, I'll call sigma bar, force per unit length in dimensional units. And one is this dimensionless rho, it's this areal density. So you see NL squared over the surface area. And there's some geometric stuff that comes into that. But those are the 2 important parameters. Now, I'm going to be quick about this. You can coarse grain this model and turn it into PDEs. You assume all your microtubules are well aligned, and you can replace individual microtubules by a density field and an orientation field. And this is work that I did a few years ago with David Stein at Flatiron. It's extremely useful as an analysis tool. And all that happens, I'll just say, is that if I introduce, just as another parameter, as another Lagrangian parameter, where microtubules are anchored, that's also a Lagrangian label for a microtubule plus its arc length to give position, then that local slender body equation just becomes a Lagrangian flow map for the microtubule bed. It's just a flow map. And then the summing through the Stokes equations turns into a Stokes equation forced by a force density that lives where the microtubules live, that green part right there. Omega sub mt, that's a what the hell is it called, a cutoff function. What do I want to call it? Is 0 in one place, 1 in another. Characteristic function, sorry. Who's in my fluids class? Okay, that's Amy Coulter. So, and so what happens is that when you write down these equations, you have this force that drives things in the microtubule mechanics. And then these are Lagrangian objects, and you have to turn it into an Eulerian force density. That means that there has to be a determinant from the flow map, the deformation tensor that shows up there. And what also shows up is this areal density. So as the number of microtubules grows, then this term becomes more and more important. Okay. And it turns out that this system has a nice energy balance equation. And then we'll get into more interesting things, but I just want to point this out. That the natural energy for this system is the elastic energy that's stored in the elastic fibers in their deformation. That's the natural energy. And if you look at its time derivative, it has an input power term, that first one, that comes from the motors performing work on the system relative to the microtubule orientation. elastic energy gets decreased. That's just grad U right there. That's just viscous dissipation, the rate of viscous dissipation. And this is an additional dissipation term that comes from microtubule drag relative to the background. And what this, it says an interesting thing geometrically. It says if you have a non-trivial state where I have a non-zero elastic energy, but I'm in steady state, so nothing's moving except for the fluid, then to have a input power which is positive that allows this epsilon to be positive, needs to balance the dissipation, then you have to have that microtubule under motor protein load is aligned with the flow that it's producing. So this is kind of self-consistent, geometric self-consistency. Okay, there's a lot of words. But you can take this model and you can do stability analysis on it. Let's say I take that PDE model and I just embed it into a cylindrical pipe. Okay? And the steady state, it turns out it has an exact steady state where every microtubule is just pointing straight in from the boundary as an orthogonal vector. Here are all the vectors the same way. And it turns out that in that steady state, if you calculate the tension in each of these, you find that the motor protein load creates a negative tension, which is compressive. So this thing is sitting there under compressive load, it's waiting to go. You already know it's going to be unstable. Once the motor protein forcing gets high enough, this has to buckle. And there's the stability diagram, and I'm going to show you a movie from this PDE model in what's called the streaming domain. So this is as a function of dimensionless motor strength rather, and dimensionless areal density of microtubules. There is a region that's called streaming, which you only access by having enough microtubules where everybody bends over as one collectively and does this. Boom. They've all bent over collectively, and they have now inclined all the microtubules so there's a canopy where all the motors are walking now across the top of the canopy, and they drive a big flow. So in this kind of model, there's an instability that leads to a large-scale flow, which is on the scale of that container. Now, we've done other work that shows that the most unstable mode of all the azimuthal perturbations that you can put onto this are the ones at m equals 0. It's a global mode where everybody just bends over.
Do you start that from some sort of random condition? Like
This one not, but just wait.
Okay.
Okay, so that's all very exciting. I can tell how excited you are. So let's go back to a three-dimensional cell. And now this is, I'm not going to tell you how we do the numerics, but we're good at this. This is a couple thousand microtubules under this motor protein load model. And now it's in a spherical cell. They're all pointing in. It's in this streaming regime. Boom. And they are all perfectly straight, and the randomness comes from the fact that we cannot uniformly coat a sphere with things. So there's asperities, if you like. And what it does is it chooses a direction around which everybody bends, and it chooses a big flow that just goes around and around.
Does the topological defect continue to get advected, or what happens?
Okay, so this is an interesting question. At the least, there's a defect here because having one of these flows and having a bed of bendable objects that are bending relative to the surface necessitates a defect. So Poincare-Hop tells you, I have to have a defect. One question is, does the defect is it anything other than the signature of a large-scale flow acting on the microtubules? The answer is no. It's nothing more than that. Though, when you see it, you always want to say, oh, there's a defect, and everybody gets excited. Maybe the defect is driving the flow. It's not driving the flow. Okay, but it's interesting. You look at it, you think it's a liquid crystalline system. It's not. Yeah.
In a real cell, you also have the constraint that all these microtubules gather at the centrosome, right? On the other side.
Oh, okay, no centrosome now.
Okay.
Centrosomes is the embryo problem. Now we're in the oocyte. All of these microtubules are nucleated from the boundary of the cell.
Okay.
I kind of jumped into another problem. If I have time, I'll come back to centrosomes.
And so here, once they bend, they remain bent?
Yes, it finds a steady state. And that's what it does, is it finds an axis, and we've cut it through the vortex. And if you cut through that vortex, it looks like a solid, primarily like a solid body rotation, David. Solid body
I'm
rotation.
looking it up.
And so, at the defect, microtubules don't have a prescribed direction of bend. It's a little more disordered. It's a stagnation point for the flow, so they tend to be more straight. What that means is they will pump fluid down the axis because they have force on them. And so actually what you see in our simulations is that there is a kind of a bitoroidal flow, where the bitoroidal part is primarily this. The bitoroidal part comes from the fact these defects, about 20% of the speed of the main flow, are creating this kind of circulation. Okay, and okay, there's the stability diagram that comes from running this big code. The stability diagram on the previous one came from just solving for a bunch of eigenvalues. It took on the order of less than a second. This one took about 3 million CPU hours. So anyway, that other one, while it's restricted, it's kind of saying what the basics are. And so in this kind of stability diagram, again, you see things that oscillate. In fact, you can see oscillations in microtubules that sit at these defects. They can be stable down here if you have low motor speed or low density, and that's the streaming state. They just all kind of bend over and collectively push. Okay, so what happens if you put it into something that looks more like an egg cell? So, as I said in the paper, and the referee didn't say a word about it, didn't even chuckle, startlingly enough. I said, oocytes are not cows. We will take an egg, Right. Who gets that joke? Anybody get that joke? It's a joke that David and I can share. So here's now 2 halves of the cell, and I should say on the previous one, you will kind of, choose an axis of this thing depending on what the conformation was of the initial microtubules and how they're placed. There's in some sort of continuum limit, it just has spherical symmetry for its initial data, so to speak. So there's some symmetry breaking instability. Now here is, again, a couple thousand microtubules that are stuck into a egg cell. And I just want to tell you what you're going to see. It's like the first little newspaper reporting. You're going to see, we call these twisters. Why do we call them twisters? We call them twisters because they have these axial flows that sit on top of the vortical thing, like tornadoes. So what you're going to see is that one of these twisters is going to form across the cell, the short direction. And then you're going to see that it slowly rotates and becomes axisymmetric. There it goes. There's the 2 defects on either side of the cell. That happens very rapidly. That happens on the collective relaxation timescale. And then your whole vortex is slowly reorienting itself, as far as we can tell, on this single microtubule relaxation timescale, which is long, so that it sits along, gives you a vortex which is lined up with the long axis of the cell. So Glenn, this is a really crappy transport flow. Now, yeah, I saw her hand go up.
Mike, so how does this depend on the local curvature of the,
That seems to, okay, I don't have a good answer for that as yet. We know most of the analytical things in spheres. I can give you arguments as to why large-scale twisters are global attractors in spheres that have to do with doing Landau expansions. of some simplified model. What I'm going to argue is that this is what you see in the later development of egg cells. So these are slow relaxations, longer than you can typically keep an egg cell viable outside of the organism. But what you can do is you can go to later stages where, in the real beast, it just keeps moving, and the cells become more and more ellipsoidal as another. And we've looked at cell shape. It's kind of complicated. That's what I'm trying to say. Except that when you're in these long cells, it becomes axisymmetric.
Is it reasonable to have these rigid boundary conditions? Presumably this
That's
flow
an interesting
changes the
question
shape
too.
of
Yeah,
the
so
egg.
maybe not. So we assume that these microtubules are static for the time course of the whole simulation. They probably, from estimates in the experiment, they're probably living for something like 20 minutes. So there's going to be some disassembly and reassembly, and they are also embedded into the cortex of the cell, which is not a rigid substrate. So they're in flows, they're being dragged on, they're probably moving around a bit, things like that. Plus, in truth, I've assumed a lot of things, partly because it gave me a model that I could analyze. So that they're rigidly clamped onto the side of the cell, for example. There's probably a lot of compliance, for kind of bending at the base. I've assumed that there's other ways in which you might generate flows, maybe through similar mechanisms that are not quite so rigidly described. But this still nonetheless gives us right types of flow speeds for a reasonable number of motors on per microtubule, things like this. So the flow speeds here are 100 to 300 nanometers per second. You could do an estimate of how many motors you need to get those types of speeds, and it's like 3 to 5 motors per microtubule. It doesn't seem crazy.
And how long are these microtubules?
That's another thing. We do from light microscopy, you can see what look like very long microtubules. They're likely bundles. We just assume that they're 20 microns because that's about the scale that you see for this dense forest, that we can't really discriminate on all the stuff in between. So we just assume 20.
In the earlier experimental slice that you showed us, there were two major vortices, and they were kind of like not the stagnation
No,
point was
yeah.
nowhere near the pole.
So in the real cell, I am certain that you do not have a uniform distribution of microtubules, first of all. I think there are probably, I'll say two things. One is from these types of twister flows, if you use them to kind of virtually reconstruct flow profiles, most of the microscopy is taking place 20 microns into the cell because they're optically dense. So you can't get in very far to see things. So about 20 microns, 30 microns, something like that in. This is where we're looking at velocity fields. And you see things that look like streaming flows, like across. You see things that look like vortices, sometimes more complicated. We can see many of those simply by sampling this basic flow at different times during the orientation that look like things that you see in the experiment. So we think a lot of the variety to see in the experiment, you're seeing one thing. You're seeing a twister, maybe it's not in its final configuration, but you're seeing a twister that's kind of randomly sampled because that's the way the experiments are done. And in fact, when we, keep an eye on the time for me if you wouldn't, Madam Chairman.
You have 5 more minutes.
5 more minutes, okay. So when we were first getting samples from this lab in Northwestern of Gelfond that we worked with, we were only seeing nice, beautiful vortices. And we were asking, well, how do you find them? And well, it turns out that they were cherry picking, and like 1 out of 10 had a nice vortex. But if you actually don't cherry pick, then you see a much wider variety, which is more consistent with random sampling of orientation of something that looks like this. Does that make sense? Okay. So Stas and I, so a lot of this work, all this work is I should say, with Stas Schwartzman, between Flatiron and Princeton. And Olenka Jain is his graduate student. He and I co-advise her now. And Dr. Chakrabarty was a postdoc in the center. He's now at ICTS. And so, but what we have done is look at later egg cells, take them out later, and then examine them. And what you find is that of the 23 that I think at between stage 10 and 12, according to that picture, that Olenka excised and looked at their flows, 22 out of 23 are axisymmetric flows. So that seems to be the stable configuration. It would be consistent with our model, and we do simulations and look at the effect of cell geometry, which gives you changes in curvature. You could ask what should happen when you have a pipe. You go to we can talk about whether there's different interesting things that happen as you do change the geometry. And the other thing is that there was a kind of a folklore in the field that these flows were handed. So it turns out they're not. Certainly not in our hands. But if you go to Olenka's data, you find that it's 23, but 9 were this and 11 were this. And, you see no sign of handedness in these flows at all. Now, a couple of other results, and then I'm going to stop. So we've done a lot more work on this. I'm just kind of telling you a couple of things that you might find interesting. One is that energy balance equation, you have different sources of dissipation and you have power input. The dominant sources of dissipation is really the drag. It's not the loss that you get from producing gradients in the loss. It's not from the shear, the background shear of the flow. It's actually the drag on the microtubules that's the flow is going by. And that comes from both examining the simulations and because you could just do scaling analysis on that energy dissipation equation by assuming you have long, thin layers. The other is that the defects do not drive the dynamics. The defects are a consequence of the fact that you have a vortex which is impinging onto these microtubules at the end that's being produced by the bulk distribution of vortices going around. To check that, we shaved the end of our simulated oocyte of microtubules. And then what you see is this thing forms across the egg as usual in our simulated egg, and then just rotates and doesn't really notice that there's no longer a microtubule defect by moving the stagnation point out of the microtubule egg. So it's a bulk dynamics. There isn't really a big contribution from storing special elastic energy or gradients into defects and things like that. Okay, now, I think I'll stop there, Alexander. Oh, let me show you a movie, because this one is weird. It's okay? And then I'll stop there. Okay, so I have like another 20 minutes. And that 20 minutes has to do with the dynamics of centrosomes. So I talked about centrosomes being these things that are nucleating microtubules and hitting motors and things like that. In those first flows where you're moving nuclear material into the middle, that's all being negotiated by two centrosomes that are attached onto these objects, the pronuclei. So they're binding on the motors, they're moving things around, and as they move things around, kind of the force landscape that it sees changes. But fortunately, the microtubules in this thing live for only about 20 seconds, so it's a very compliant object to changes in geometry. So we have models of that and investigating that. But I just want to show you a movie because it's creepy. So these are two centrosomes. Now here they're not nucleating, we just have permanent microtubules, but it happens this way anyway, at least in our models, we think in the cell, that are competing for, these are dining motors on the surface.
Ooh.
And what they're doing is they are binding onto dining, which is pulling on them, and they're moving apart. Why are they moving apart? That's because when they are close to each other, they are competing for motors in this regime, and the motors that are further away, they don't compete for, so it creates a force imbalance. So these guys are naturally repulsive to the scale of the objects. So they will move apart because they're competing for the things that are moving them. And it turns out that this kind of model, let's see if I can get it, we have used in other papers to predict what should be the length of the spindle in C. elegans because the two centrosomes are competing for motors. and they will move themselves in a certain egg shape to a certain distance. So so it turns out there's a whole game you can play with multiple centrosomes inside of a cell competing for motors. And what it does is it reproduces the Thomson problem of having platonic solids, some of which are stable and give you the energy minimizing solution to the Thomson problem, and others where the platonic solid is not stable. And it's the same set of platonic solids which is not stable. So anyway, I thought this was fun. There's a physical review research, PRX life passed, so okay. Too theoretical. Anyway, I'll stop there. Thank you.
Questions?
So, if I understand right, then these vortical flows are a consequence of the cell going about its business. So,
Yeah.
And do some of the vortical flows you showed us tend to mix things up, like the bivortex, and others don't mix things up, like the twister. So, do cells rely on these vortices to keep themselves nicely mixed?
So the other thing that has to happen, and this is still a mystery to me, an open question to me. Other things that need to happen is that you have yolk granules that come into the cell through endocytosis inside the cell, and they have to get suspended into the bulk. So it's like a big multi-body problem. So I think it might have to do with, at least in some ways, with suspension of things. Yeah. But that needs to be tested. And
You mean against gravity?
no, moving them from the boundary and distributing them through the bulk. That's a suspicion. That's the answer I give to this question.
Glenn?
So I don't know if I'm asking the question the right way, but having all of these filament microtubules bent, each bending one microtubule takes a lot of force or energy, and each motor only creates a few piconewtons, and it's due to so it's because of coupling through the fluid field. So is it the case that you're basically using the fluid flow as a way of accumulating work done by the motors?
Yes.
So is that, I don't know, a good general method to use fluid dynamics to harness
Okay.
motor energy?
The usual thing is I'm going to answer the question you didn't ask. But okay, you can do the estimates. Let me just say that. Right. So assuming that this motor is exerting 5 piconewtons of force, and there's a certain speed that you need to have, how many motors do you need for microtubules? Somewhere between 5 and 10, 5 and 8. So, like I said, it doesn't seem crazy. That's assuming a payload of a certain size as a sphere, and that may also be not true. The one thing I want to say, which is not what you asked, is that this model is basically the model for, from the surface, pushing a fiber into the fluid. So you push a fiber into the fluid, that pushes the flow out. So these are fountain flow problems. And then they will buckle because they're under compressive load from the viscous stress. And then they will buckle together because of the viscous coupling. That doesn't necessarily have anything to do with microtubules and motors. So I've been wanting somebody to do some sort of lab synthetic version of this. So for example, let's say that you want to produce a bunch of actin tails. Right? You have actin tails on the back of the bead, they propel this guy back. The actin's a drag load and it's polymerized in the front, the whole thing kind of moves along. Imagine that you take what do you coat? There's a protein.
Yeah, ARP2-3 complex.
ARP2-3 complex. Say you make little dots of it on the surface, like an array of them. They will start producing actin tails moving, I'm guessing, away from the surface. And these guys disassemble at the backside, so it gives them a finite length. And they will act like the mathematical model where they're pulling fluid up and they're under compression. So I think that these will create a buckling that will then drive a flow plane. I would love to see somebody do gas experiment like that.
Yeah, you should check out what the Grenoble people already did. Maybe they sort of did that already, but they didn't talk about the fluids.
Oh, is that right? Okay.
But I don't love that. I don't love this explanation that it's using the fluid to, because there's no inertia. The problem has no inertia. So
Well, okay, so there's nothing stored in the fluid?
yeah, there's nothing stored there.
There's
So it's not like
nothing
it's not accumulating force.
Yeah, maybe I might be agreeing too readily. I'll let you
Yeah.
guys argue about it. But what it does do is give you this instantaneous
Yeah,
communication.
it makes sense that it communicates, but it's not. I don't think it's integrating.
If it's not in
Work.
the fluid, then I think it's in the
Yeah, I don't
filament,
know.
in the bend,
Yeah,
because it's more force
Yeah,
than.
it is
you know.
integrating forces that are coming from microtubules. That's exactly what it's doing.
Yes,
Yeah.
but not over time. It's integrating it over space.
Yeah.
Because you have no inertia.
That's true.
Any more questions here? Adam, you have your hand up or
Oh.
no?
Just hanging out.
Yeah. All right, so
Okay,
let's
thank you.
sing back again.